Now let's kick off our first panel, the life science panel. Let me invite Professor Li-Chun Law, the session chair, to the stage, but let me briefly introduce him. He's a professor in biology, expert in neuroscience. He's a member of National Academy of Sciences. Li-Chun, please.
Good morning, everybody. It's my pleasure to introduce Professor Chuan He, my fellow alumnus. And Professor He is currently John T. Wilson Distinguished Service Professor at the University of Chicago and an investigator of the Howard Hughes Medical Institute. Professor He's pioneering work on elucidating the mechanisms of RNA methylation and its many biological functions has earned him many awards, including the prestigious Wolf Prize in Chemistry in 2023 and a belated election into the National Academy of Science a few months ago. Please join me in welcoming Professor Chuan He for a keynote speech.
Thank you. Well, thank you, Li-Chun, for the introduction. I'd like to thank AASF for this opportunity. This is the life science session, but I started as a chemist. I was trained as a chemist and started my career as a chemist. I decided to switch career in 2008 because I was puzzled by the complexity of human. Now we each have tens of trillions of cells, 200 different cell types. We have amazing functions. We can walk, we can think, we have a conscious, we can self-repair our wounds. But all of this complexity come from one fertilized egg. We all come from one fertilized egg. In a single cell, in the genome, which is only 3 billion base pair long, store all this complexity. How does that work? So that was actually something got me really interested. Back in 2008, I decided to explore epigenetics. Now, for decades, scientists have shown that genome is not just a single sort of plain line. It can be modified. For instance, our DNA can be chemically modified. These modifications actually encode additional information. If you have a modified DNA, that will suppress gene expression. If you're less modified, that will be actually activate gene expression. You can actually titrate this almost in the gradient. Now, the proteins that package DNA, the histones, they're also chemically modified, and that encodes information to tune gene expression as well. So now we do not need to view genome as a line, right? Analogy will be the key, right? If it's a plain key, there will be no information content, but because of these T's, right, a short length can carry enormous amount of information. Now imagine this is a 3 billion base pair long or 3 billion building blocks. Some of those can be reversibly modified. The combination gave us the complexity that eventually represent the complexity of trillions of cells. Now, when I started in 2008, I asked a simple question. For decades of work, people show that the DNAs and histones can be modified. What about RNA? RNA modification has been known for a long time, and we asked, could RNA modification also represent the sort of a T's of a key, also present sort of the complexity that impact the eventual gene expression? So we show in 2011, indeed, this methylation can be reversed, and this process is critical to physiological processes such as embryo development. The mark we worked on, this is N6-methyladenosine, turned out to be a very interesting one. This was discovered back in 1970s, and is the most abundant modifications are messenger RNA. Remember, our genetic information go from DNA, messenger RNA, to protein. So in all of us, every messenger RNA, in average, carries 3 this methylation. It's also abundant in plants, in other eukaryotes. the community recognized this is important back in the 70s, but because of lack of molecular biology, all the fancy technologies we have today, for over 10 years, it's kind of died out because it's very hard to study. Now, we discovered this mark could be reversed by two demethylases, FTO and ARCBH5. Now, keep this in mind. The rest of the talk will be on these two demethylases. So human or mammals have two types of demethylases that would remove the mark, right? And this activity is critical, essential to embryo development, to all of us develop into an embryo, eventually to a baby. Now, the methyltransferase turned out to be complicated, and my lab and lots of other labs discovered, characterized the binding proteins. These were evolved to selectively recognize the methylated RNA. This way, we can actually recognize the methylated RNA, hundreds, thousands of them, for stability regulation, translation regulation, and regulator localization. So this turned out to impact almost every process of life, and it's been taking off. Started from 2011, the first paper. Nowadays, thousands of papers. This affects development, stem cell differentiation, all kinds of human diseases, immunology, and learning, memory, and neuronal functions. What I'd like to share with you today is something different. While we were working on this messenger RNA methylation, starting in 2018, 10 years later, we realized in addition to messenger RNA that carries genetic information eventually translate into proteins, there are these regulatory RNAs on chromatin that are also methylated. And we wondered why, right? These are non-coding RNAs, they're not coding anything, they're just hanging around on the chromatin. And turned out if we remove the methylation, we dramatically affect gene expression. Now, this is an experiment we did. This is the label of the nascent transcription, right? So how much RNA is produced from the chromatin. As you can see, if we get rid of methylation, we delete methyltransferase, we see dramatic upregulation of total output of RNA. And this is another experiment. We look at the chromatin openness, right? Chromatin opens, gene expression close, gene suppression. As you can see, we label them with fluoroform. We look at all those sites that are open. And again, the depletion of these non-coding RNA methylation, we saw dramatic upregulation, opening up of chromatin. So that led to actually discoveries. We realized where some of these non-coding RNAs from others' work, they're critical to assemble transcription-active complexes, drive gene expression. They are regulated through methylation. Now, these methyl groups, just like in messenger RNA case, they can be recognized by the binding proteins that specifically recognize the methyl group, and they can recruit additional factors to regulate the local chromatin state and regulate transcription of that particular gene. Right? This turns out to be a general sort of theme in biological regulation. These writers not only put methylation on messenger RNA and do all the things I just introduced, they also put the right methyl group marks on these chromatin RNAs, and that leads to recruitment of different reader proteins or binding proteins, and this regulates chromatin state and transcription. The demethylase turned out comes in to reverse this process, and that function is essential to embryo development. So from here, we can add another dimension to what I told you. Remember, we say 3 billion building blocks. There are different marks on the building blocks that give you complexity. Now, this building block, a block can be transcribed into RNA, and there's additional modifications on these RNAs that add another layer of complexity. So that's my introduction. I don't believe in this podium anybody has talked about plant biology. I'm going to transition to plant biology because you've heard enough about human health, biomedicine. We work on all those. I'm going to actually transition to plant biology. Now, methylation not only occurs to humans, to animals, to mammals, also occur to plants. And it's prevalent in plants, and I will argue may have more important functions in plants. Here's just the one example. This is the work we participated. We did not lead this work. This was done by somebody else. They look at very interesting one question. In cucumber, this is Y-type cucumber. This is today's cucumber you get from supermarket. They're long because of domestication, right? We select mutations. Eventually, somehow, they grow. We can eat them. And this group asked what leads to this. They screened thousands of genes, variants, identified one mutation that responsible for converting the Y-type cucumber to today you grow in your backyard. That mutation is on the messenger RNA, wiped out a single M6A methylation site. That methylation regulates translation of that single gene. If you bring that site back, you shorten the cucumber length. I just give you one example. One single site stands for cucumber length. Right? If anything you learned today, this is one. Now, we've been interested in a different question for many years. In mammals, there are two demethylases, FTO and ARCBH5. In plant, they have 5, 7, 8 different demethylases. Many of those, but they all belong to ARCBH5, not FTO. We wondered why. In fact, these plant demethylases have all kinds of interesting properties. They can regulate early flowering, vegetarian growth, etc., etc. But we asked, why not FTO? Why they only have ARCBH5 homolog and they have so many? So out of sort of word experiments we did, it was coming out of a discussion when I was visiting my former postdoc and a plant biologist. And we decided, well, let's test this out. Let's put a human FTO into plants and let's see what's going on, right? I forgot about this. A year later, they come back, told me, Professor He, here's the data. When we express human FTO in plants, we really drive plant development. Here's the rice. We increase biomass by 2-folds and we increase yield by 2-folds in greenhouse. In real field test, 20 to 40% of yield increase. And this is complicated. It drives plant development. You clearly see improved root development. You clearly see consistently 20% increase of photosynthesis. And these plants are very robust, resilient to various stress. Here's the drought resistance. They're more resistant to drought, heat, and other stresses. Right? Very complicated system. So this was great news, right? And we asked about other things, right? This is for my biological colleagues down there. We showed how this works. It's going through chromatin regulation, opens up the chromatin state, just like what I shared with you. The entire transcriptome uplifted. So the entire output of transcription is uplifted from this chromatin regulation. And that leads to more cell splitting and cell proliferation. This is the tip of the root, and which drives root development. And this also impacts the stem cells on the tip, and that generates more telomeres or more biomass. Asians are like rice, but here's for others. This is a potato. This is a tomato in my lab. We occasionally eat them. They're fine. You can see the yield, and here's alfalfa and grass. Right? You can clearly see increased yield, biomass. Now pay one attention. These are greener compared to the control, right? There's 20% increase of chlorophyll in these plants. This is great, but this also generates a big problem, right? This is transgenic. It's okay in this country, but in the rest of the world, that's a no-no, right? Even in this country, a big problem is you don't want a human gene in your plants, right? No, that's a big no-no, so forget about it. So we went back and asked, well, we saw this. How can we take advantage of this? And maybe we should take a look at the basic science I shared with you. When we overexpress FTO, we see this really interesting phenotype. But if we overexpress ARCBH5, human ARCBH5, there's no such observation. There's no change. If we take a plant ARCBH5 homolog, same thing. They're just staring there, look at you, no change. Eventually, experiment we did over 15 years ago came back to remind us what we might have been missing. Now, here's the imaging of FTO. As you can see in mammalian cells, it's everywhere in the cell nucleus. But if you look at the ARCBH5, it forms this puncta. And we went back to the ARCPG5 sequence. For biologists, this puncta typically indicate they have this so-called intrinsically disordered regions or low complexity domains or whatever you call them for the rest of the audience. There's just floppy spaghettis, but they have function. They stick with other proteins to aggregate, right? And that's the reason they form this puncta. And these typically have biological function. We said, well, if this is the case, let's delete this. There's an N-terminal and a C-terminal. Let's delete and see what's going on. If we delete the N-terminal, they still form this puncta. But if we delete the C-terminal, now actually they behave like FTO. They distribute across cell nucleus. And when we analyze the chromatin state, the Y-type did not activate chromatin state, as I told you many times. But once you get rid of C-terminal, you got rid of this is flexible spaghetti. It goes around and activates chromatin state. Feels like we turned this into a FTO-like protein. Now here's the full length. This is the truncated. They have some overlap, but they work on distinct substrates now. The full length on messenger RNA, the truncated started to work on chromatin state. Okay. So what we learned, these ARCPG5 Y-type have these floppy domains to restrict them on messenger RNA, yet the FTO goes around, work on these non-coding RNAs on the chromatin to tune the overall output of transcription. Now, if we get rid of this C-terminal, we convert the Y-type into FTO-like demethylase. Right? And here's, again, for the biologist sitting on the first row, here's the data. This is the histone marks. These are the histone modification stats for gene activation. Once we get rid of C-terminal, you can clearly see increase of these marks and recruitment of transcriptional factors indicating, yes, these indeed work. They recruit proteins to activate gene expression. Then we realized that this may be a strategy we can engineer plan ARCPG5 homologs, right? We can start with mammalian systems, identify features that allow us to activate chromatin, and we can then test this in tobacco leaves, white tobacco leaves, because this is the model system. We can do very rapid experiments. And then we test in Arabidopsis, a model plant which grows in 3 to 4 months. We learn in the real plant what's going on. So we overexpress FTO and human ARCPG5 with C-terminal deletion in embryonic stem cells or in mammalian cells. Indeed, they activate chromatin, as I showed you. We then put this into tobacco leaves. As you can see, FTO activates chromatin. You see all the fluorescence increase. This indicates more open chromatin. The ARCPG5 did not do that. The N-terminal truncation, no. But once we got rid of the C-terminal or N and the C-terminal, we convert this, behave just like FTO. They activate chromatin in the tobacco leaves. What about in the real plants? With FTO, we drive the root development. This is the mutant, so get rid of the demethylation activity. That's our control. But if we put ARCPG5 full length, we did not see this promotion of root growth. Once we remove the C-terminal domain, we overexpress this human demethylase. We drive root development. And this is the one that we got rid of both N-terminal and C-terminal. What about, well, this is again for the folks sitting on the first row, same histone marks, but now we did this in plan and convincing ourselves, indeed, all these marks that sort of represent transcription activation are elevated. Obviously, we went on to look at the plan ARCPG5 demethylases, right? There's Arabidopsis, our model plant, but also rice. They all share similar structures. They all have the C-terminal floppy ends, just like human ARCPG5. And biochemically, we can convince ourselves they're indeed a demethylase. Here's the FTO. We can see the demethylation of a model substrate. This is the green algae. It also has FTO homolog. It has biochemical activity. This is Arabidopsis. This is the rice demethylases. They also persist activity. Not as high as human FTO, but they are active. So when we put this into mammalian systems, we activate chromatin, the full length, just like a human RNA 5 homolog, full lengths are not very active. Once we get rid of the C-terminal domain, they activate gene expression. And in tobacco leaves, this is the rice demethylase. Once we truncate the C-terminal domain, they started to activate chromatin. And once we put into Arabidopsis, they promote root growth. So they started to behave like human FTO. We eventually tested real crops. This is in rice. Rice is easier to test in these systems. You can get results back in the year. So again, when we use the human ARCH5, the full length, we did not see much difference. But once we get rid of the C-terminal domain of the human ARCH5, as you can see, there's actually more tillers on this plant. And if we put them together, analyze the yield in greenhouse, we see about 30 to 40 or 50% increase of the yield, and there's an increase of photosynthesis. Preliminary results in the field test, put this as a grain of salt. We need a lot more experiments, but in the real fields, we also saw about 20% yield increase, and we consistently see increased photosynthesis. So we've gone from essentially this is actually the rice demethylase, right? We started with a human FTO. We move on to human ARCH5. Now we move on to rice ARCH9B, which is a ARCH5 homolog. Now this is the rice protein. When we get rid of the C-terminal domain, we can promote growth compared to the Y type. That's wonderful, all right? Now we seem to solve our problem. We have the human genes in plant promote growth, which is not good. Now we have a plant protein. We cut off the C-terminal domain. We put back into plant that promotes growth. We have gene editing these days. One of our panelists is a CRISPR experts. We can easily come in and use gene editing to introduce a stop codon, introduce one mutation, eliminate the plant's own C-terminal domain, right? Convert the plant Y-type gene into a truncated version. And so forget about GMO. In this case, in principle, we should be able to generate FTO-like activity. That was precisely what we tried, and very preliminary results. This edited rice also gave us increase of biomass and photosynthesis. So that was really an interesting exercise from very fundamental basic research discoveries led us to solve this problem. And these demethylases are conserved in all plant kingdoms. We're currently testing a variety of different plants: rice, tomato, potato, soybean, maize, cowpea, cassava. All right, we're making up hundreds of these edited traits and want to follow what's going on. Now, that's meant to solve food security problem. Food security is never a problem for US. It never will be a problem for people sitting here in Colorado, right? We don't worry about food security, right? It's Africa's problem. But everybody worry about climate change, right? I was in Chicago 2 months ago. We have all these smokes coming in from Canada, right? It's like wildfire, everything. The planet is heating up. We have to change our agricultural behavior. Now, I will argue these were probably the strategy to help face that challenge. If you look at these plants, this is a tobacco system. They grow better at higher temperature, right? They're more resistant to stresses, right? Here's Arabidopsis. This is the Y-type Arabidopsis. It's 37 degrees. 75% of the plants wiped out because of heat stress. But our engineered version survived. 90% will survive. So this is an interesting strategy. Not only increase the yield, but also increase the resilience to stress. And I think moving forward, this could be eventually applied to agricultural practice and hopefully will benefit one of us. Now, I'd like to stop here by thanking student postdocs. I started as a chemist, become a biologist, and now I'm living in Chicago, become a part-time farmer. So I set up a plant biology lab, and these were 2 postdocs who did the work. And I really want to thank these foundations, the Pritzker Foundation, the Gates Foundation, Harborview, Orthodox Philanthropy. They sort of put a lot of trust into our work and really helped drive this work forward. This is my disclosure and my collaborators. Thank you very much.